Okay, good morning, students. So, in continuation with the previous lecture, today we'll continue with culture media. So, before we delve deeply into culture media, it is imperative to know the meaning of culture. So, in general terminology, culture means a tradition or some practices associated with a particular group of people or population from a particular region or area. But it's different in microbiology. In microbiological terms, the word culture means to grow or to isolate. So, what do we grow and what do we isolate? We need to grow or isolate the microorganisms, the infectious microorganisms from the patient's sample. For example, if a patient is suffering from urinary tract infection, so naturally he'll be having bacteria in his urine sample. We need to separate the bacteria from his urine sample. This is what is called culture of bacteria, that is growing or separating the infectious agent from the patient sample in order to identify what the bacteria it is, and also to check the antibiotic susceptibility testing, that is which antibiotics, which drugs or which medicines are going to be useful for the treatment of that particular infectious syndrome or disease. So what is the use of culture media? As I said, it is required to isolate or separate the bacteria from the clinical sample. Constituents of culture media, what is the culture media made up of? Basically, it is made up of water and electrolytes. So water and electrolytes, they serve the purpose of growing the bacteria or giving nutrition to the bacteria so that they can grow, multiply and form colonies which will be visible to the naked eye. The morphology of the colonies can be identified, can be checked in order to identify the bacteria. Apart from water and electrolytes, the culture media also have peptone. So what is peptone? Peptone is nothing but a mixture of partially digested proteins. So it's a small unit of protein and it can be obtained from beef, it can be obtained from heart muscle, it can be obtained from casein or fibrin. And how do you obtain peptone from these substances? Usually it is done by digestion with proteolytic enzymes like pepsin. So peptone, it consists of smaller units like proteases, amino acids, inorganic salts like phosphates, potassium, magnesium, and accessory growth factors like nicotinic acid and riboflavin. In order to make the culture media solid in state, we have to add one more constituent to it, and that is agar. So agar is nothing but an agent used to solidify the culture medium. It is prepared from the cell wall of variety of seaweeds. So it consists of long chain polysaccharide and a small amount of protein like material. It also has long chain fatty acids and little bit a small amount of inorganic salts like calcium and magnesium. So this agar is preferred over gelatin. What is the reason for preference of agar over gelatin for solidification of the culture media? Because agar is bacteriologically inert, it melts at 98 degrees Celsius and usually it solidifies at 42 degrees Celsius. It does not add any nutritive value. This is an important point. Agar on its own does not have any nutritive value. That means it does not provide any nutrition to the bacteria. Whereas gelatin is liquefied by a number of bacteria. Concentration of agar, there are different concentrations that can be used. In order to make a solid culture medium, we have to add 2% or 2% concentration of agar powder. In order to make a semi-solid medium, we have to reduce the concentration of agar. We have to use 0.5% of agar concentration. There are some bacteria which show swarming growth. That means they spread away from the area where the streaking was done on the culture media plates. So that type of growth or spread is known as swarming growth, and it is particularly shown by a bacteria named Proteus mirabilis as well as Proteus vulgaris. Now, in order to inhibit this growth, inhibit this swarming growth or spread of the colonies over the plate, we have to increase the concentration of agar to 6%. So basically, students, you have to remember that there are three different concentrations of agar that can be used for preparation of culture media. Usually, if we need to prepare a normal solid culture medium, we use 2% agar. In order to make semi-solid agar, or in order to make semi-solid culture medium, we add 0.5% of agar, and in order to inhibit the swarming growth of Proteus species, we use 6% agar concentration. There are different culture media which may have different constituents, like meat extract, which is highly concentrated meat stock, usually made from beef. It contains protein degradation products, inorganic salts like calcium and magnesium, carbohydrates and growth factors. Some culture media also have yeast extract, which is prepared from washed cells of baker's yeast, and it contains amino acids, inorganic salts like potassium and phosphates, and carbohydrates. Some of the culture media also have malt extract, which consists of maltose, starch, that is carbohydrates, dextrin, glucose, and 5% protein products. There are some specialized or enriched culture medium. Now, what is enriched culture medium? Those culture media which are enriched with extra nutritional sources. So some extra nutritional sources like blood or serum will be added, and then then the culture medium becomes an enriched culture medium. So the most common enriched culture medium that is used in microbiological investigations is the blood agar, where we add sheep blood. So the important components of enriched media and these blood and serum provide extra nutrition to the fastidious bacteria. Now comes the question, what are fastidious bacteria? Fastidious bacteria are those bacteria which, apart from usual or the basic culture medium, require extra nutritional source for their growth and multiplication. So usually this is provided by 5 to 10% of sheep blood. We can also use horse blood, but human blood should be avoided as much as possible because of the presence of antibodies in it and some inhibitory factors. Blood should be collected aseptically, and it should be rendered non-coagulable by defibrillation or by adding oxalate or citrate. Serum is sterilized by filtration after collection. So now we come to the classification of culture media, which is very important, can be asked for 5 marks as a short essay in your theory exams. So based on consistency, the culture media can be divided into a solid culture medium, it can be a semi-solid culture medium, or it can be a liquid culture medium. This is based on the consistency of the culture media. Based on the growth requirements, the culture media are classified as routine laboratory media, which are prepared from nutrients such as aqueous extract of meat, peptone, etc. Or it can be the second classification, second group is defined or synthetic medium, which includes simple synthetic medium and complex synthetic medium. Now, examples of routine laboratory media are the simple or the basic culture medium, the enriched culture medium, enrichment broth, selective media, differential media, transport media, and anaerobic media. What is simple or basic culture media? These are the media which are very simple and basic in nature without any added extra nutritional value or sources. So the most common example for simple or the basic culture medium is the nutrient agar, which is nothing but agar powder in nutrient broth, which consists of distilled water and peptone. Enriched medium, as we have already said, enriched medium are the basic media added with extra nutritional source, either serum or blood. So example we already have seen, blood agar is an example of enriched medium. Enrichment broth. So we'll go to enrichment broth here. Yeah, these are the liquid media which are added with some inhibitory agents which selectively allow certain organisms to grow which are of our interest and inhibit others in which we are not interested. So important for isolation of pathogens from clinical specimens which also contain normal flora. For example, the stool of a patient and the sputum of a patient have lots of normal flora in them, which we are not interested to grow. So that is why in order to isolate the pathogenic organism or bacteria from such specimens, we have to use enrichment broth, which has a particular characteristic feature that it inhibits normal commensal bacteria and allows only the pathogenic bacteria or the target bacteria or the specific bacteria which we are interested to grow. So examples of enrichment broth, we have tetrathionate broth, which is exclusively used for isolating Salmonella typhi. We have gram-negative broth for isolation of Shigella species. Also, we have selenite F broth for isolation of Shigella. And then for Vibrio cholera isolation, we use enrichment broth, that is alkaline peptone water. So the rest of the culture media we'll see in the next coming slides.